Charlie, thanks for coming.
Sure.
And Kim Kirkwood, who is in the OR today, unfortunately. So let's just get started. My project is called Risk Stratification of Pancreatic Cystic Neoplasms using protease activity. So these are the objectives for the talk. So we'll start with introducing you to pancreatic cystic neoplasms. So what is it? Everyone's heard of pancreatic cancer. This is kind of something a little bit different, but can eventually lead to pancreatic cancer, and that's the big problem. So these are the main types of cystic neoplasms that we're focusing on. Many of you have probably heard of pseudocysts and come after acute pancreatitis or trauma, things like that. But there's a couple that we worry about, which are right here. So mucinous cystic neoplasms and IPMNs, and there's two types of these, main duct and side branch. And there's also another one that's called mixed, but it's kind of just a mix of these two. There's one more called serocystic neoplasms, and this is something that we don't worry about because they're basically all benign. So it's really these. And this is something that has been increasing in prevalence over the past 20 or 30 years, and we don't really have a good explanation for it. Some ideas are that we have better cross-sectional imaging modalities now that are causing these to be increasingly diagnosed incidentally. And that's actually how they're most commonly diagnosed incidentally. They're not always symptomatic. So one study actually looking at like 24,000 MRIs, excluding patients that were symptomatic and didn't have a history of pancreatitis. 13.5% of these patients ended up having cysts on their MRIs, just incidental findings. So a lot of patients have these. And then this is another study, one of the biggest studies that actually formed the consensus criteria for managing these pancreatic cysts. Did a systematic review of each type of these cystic neoplasms, and these were the prevalences of actually having physical cancer in the resected specimens. So main duct IPMN, you can see, has the highest prevalence of cancer, with 57 to 92% of resected specimens having cancer. Branch duct and mucinous cystic neoplasms had a little bit less, 8 to 33% and 6 to 46%. And then from this study came the Sendai criteria, which is really the consensus guidelines that direct management of these. So why can't we just, if there's that high of percentage of cancer in these neoplasms, why can't we just take them out? Well, many of you have probably seen pancreatic surgeries, but it's no small surgery. Especially if the neoplasm is located in the head of the pancreas here. A lot of you have probably heard of a Whipple or seen a Whipple. But you basically, because of the blood supplies, you have to take out basically everything around that area, including the duodenum, distal stomach, and the proximal jejunum. So it's a lot. And this is from one study that looked at just the rates of morbidity and mortality from any pancreatic resections. So whether it was just a distal resection or a Whipple or a total pancreatectomy. And these were some of the percentages they came up with. So almost a 50% chance of having like a significant complication. And then almost a 5% mortality rate. So pretty high for just taking these out. And the problem with these is that not all of them progress to cancer. Some of them will have it in them, but not all of them. Some of them will just remain benign forever. So how can we figure out which ones will progress and which ones will not? So the good thing about pancreatic cystic neoplasms in particular is that the fact that they are cystic means that it gives us a unique ability to sample the fluid that's inside of it. And this is done through endoscopic ultrasound. I don't know if any of you have ever seen endoscopy, but this is a little bit different in that they have an ultrasound probe on the end of the endoscopy scope, and it actually looks through the stomach or the duodenum into the pancreas, and through that, you can see a little view of it here. This is the cyst, and this is the pancreas. This is probably a vessel. You can see the needle you can do FNA biopsy through that. And this allows us to sample the fluid. And these are some of the tests that are routinely done on that fluid. So CEA is a tumor marker, but it's actually used more as a measure of mucinous characteristic. Amylase, which is kind of used to more differentiate from pseudocysts. It's usually really elevated in pseudocysts. And then cytology, just the same as any other cytology looking for dysplasia. And the problems with these is that they're really non-prognostic and can kind of point you like serous versus mucinous, so it can be a little bit helpful. And cytology is actually very sensitive, very specific, but very, very non-sensitive, because a lot of times you won't get cells in the aspiration. So if you have highly dysplastic cells in it, you know that it needs to come out, but if you don't get anything, it doesn't tell you. So some of the background that has been done that really drives my study is the Ratte et al. They recently published some work on this SPINK1, which is a trypsin inhibitor, and they showed that in high-risk specimens, so either highly dysplastic or already invasive cyst fluid, that levels of this trypsin inhibitor were increased in malignant specimens. And so this is really great because I'm looking at protease activity. So it's showing that we should be able to expect differences in the specificities and activities because of this increased inhibitor. Key et al. Did a really extensive proteomics study on cyst fluid, and they showed that there was increased carboxypeptidase activity in these higher risk specimens. And that is really important for some reasons that I'll get into when we talk about the methods. Something unique to the technology that Anthony O'Donoghue is one of my other works in Dr. Craig's lab, is helping me out on that. He developed this specific method that we're using, and he demonstrated that it can be done on a mouse cell line in the cell secretions, and he demonstrated that it works. So our hypothesis is really that proteolytic profiles and the specificity profiles of these specific cyst fluids will be significantly different depending on their respective malignant potential. So how do we do this? So what we have done is we've already established a prospective biorepository of cyst fluid. Patients are basically taken when they come in either for surveillance or for actual resection of these cysts. The endoscopic ultrasound is usually done for surveillance, and the cyst fluid is sampled then. And then if they are chosen for resection, then we will aspirate at the time of surgery and bank the specimens. And then we have access to all of their imaging and pathology if the specimen is resected. So we can basically then stratify based on the Sendai criteria, based on all the lab values and imaging, and put them into these three categories, like totally benign, dysplastic, and already invasive or malignant. And then the two major technologies that we're using are FRET peptides and the MS-PMS technology, which I will now explain. So, yeah.
So, go back. Ones that were dysplastic, do you have follow-up data on them to see if they progressed to cancer?
We do. We're following them all. So if they do end up progressing and then having a resection, then we can have that data as well, and the histology.
So you have some dysplastics that didn't progress and some that did, and then we'll be looking at their profiles? Ideally,
yeah, we do. I don't think right now we have any that have actually progressed. I think right now we just have either people that had invasive disease and resections, and then a lot that are that have not.
I have one more question.
Yeah.
So I don't know anything about this, so I don't know how this question will go, but are there people who get identified as having a cyst, incidentally, and then they're followed, but you wouldn't biopsy them, or they wouldn't be included in your study, maybe because it's like you think it's not worth it, or it's
Sure.
dangerous to biopsy, or you're afraid of seeding?
Mhm.
Like, is there a cohort that would be excluded?
Actually, in our study, we're really only including people that are going to have the endoscopic ultrasound or resection.
Okay.
So only people that are going to be where they're going to get cyst fluid on.
And so, okay, so they would already get cyst fluid based on standard of care.
Yeah, it's basically one of the main things in the Sendai criteria is size. So a lot of the branch duct IPMNs are pretty small, and they won't actually be recommended for endoscopic ultrasound.
Okay.
So they'll just follow those with either MRIs or some other CT. So they wouldn't be included until it actually was big enough to warrant aspirating it.
Got it. Thank you.
The one thing, you're wondering whether maybe even the endoscopy itself might tip the balance and skew the data.
A little bit, yeah.
That, yeah, how do you control
Yeah.
for that? Yeah,
That's
it's a reasonable
It's-
point.
Yeah. It's a tough
I
issue.
see what you're asking. And the control of the control. It's something we should keep in mind.
Yeah, there is kind of a tough. There's actually a kind of a debate in the literature about these endoscopic ultrasounds in general and whether it could possibly like see
Yeah.
through the wall and stuff like that. But I think overall the consensus has been that it's worth the risk for the patients that warrant it. So PrEP peptides, what they are is basically a custom designed peptide. And then ours are 8 amino acids long, so each one of these represents an amino acid, and we can design them in basically any combination we want to. On either side, there is a fluorophore, a fluorescent tag, and the other side is a quencher. So as long as that peptide is intact, it will not fluoresce. And we can add whatever specimen we're adding. So in our case, it's the cyst fluid that has like a whole plethora of proteases. And if the sequence in that, one of the sequences in that peptide is targeted by one of the proteases, it'll get cut, and then it'll show fluorescence. And so through this, we can kind of have a measure of protease activity in the sample. And one of the things we're using this for is kind of to screen it for the mass spec assay that I'll show you next. Because if there's too much protease activity, then it'll basically eat all of our substrates up for the next, for the mass spec assay. So we have to, this is a good way of screening them first. And also this allows us to, it's a very fast, it can be done in less than an hour. So it's potentially a way of, we can design these to target either be targeted by specific proteases or just to be targeted by specific specificities of a cyst fluid. So one cyst fluid has a specificity for to cut a certain sequence, we can design a peptide to show that. And that'll be a little more clear. So this is kind of the meat of it. This is the main method that we're using. It's called multiplex substrate profiling using mass spectrometry. And so basically what this is like the PrEP peptides, it uses substrates that are 14 amino acids long and do not have any tags on them. So they're just, and there's 228 of them that were designed basically as a library to cover a really diverse specificity. And one way, I don't know if this is a good analogy, but one way to explain it was if you were going to go bird watching in a specific park and you want to be able to identify any birds that are in that park, you need a book that has pictures of all the birds that are in that park. So that was one way I came up with to figure that out. And so that should be able to identify a really, really diverse set of proteases. So what we do is we get the cyst fluid either by US or by aspiration during the resection. And right now we're controlling them all based on protein activity. So I find out how much total protein is in each sample diluted, so they're all run at the same protein concentration. And then we add it to this peptide library. And then once I do that, so the proteases are now starting to chew up the substrates, and I take out time points. So at 15 minutes, 60 minutes, 4 hours and 21 hours, I take out a little bit and I basically put it into really concentrated acid that kills all the activity in there, and then I can run them on the mass spec. So you can see right here, this represents a peak. Each of these is a substrate, a whole substrate. And then this is, you can think of this as one of the time points. So say after 4 hours, now we see all these little time points. Back. All right. So you can see all these little time points, and these represent fragments of the substrates that are popping up. And the good things about this is that it really allows us to get a global picture of the protease activity. So it's looking at all the proteases that are in this sample. You don't need to hone in on one by more difficult techniques. And then another thing, I mentioned the carboxypeptidase activity. One benefit of this is that you can specifically look at carboxypeptidase activity because there's no need for tags on these carboxypeptidases. If you remember from biochem, carboxypeptidases eat it from the C-terminal, so they start at the side. And if you have a tag on that, it's going to interfere. So we don't need tags on these, so it actually gives us a great look at this activity.
Are you able to identify the activity by which substrates are cleaved or by the pattern on the
Both. So we will take all this information and basically put it into a program that reassembles all the cleavages and figures out which substrate each cleavage event is from. And then it'll basically give me a giant spreadsheet of all the cleavage events for each substrate and at what time points they happened. And so this is, yeah.
I was just curious if there's any difference between the structure of your peptides versus the natural structure of the peptides that might be in this cis fluid, and does that impact the ability to actually have cleavage?
Oh, you mean just like things that have already been
Right.
cut in the specimen itself?
Mm-hmm.
I think this is, so when we compare, basically when I'm putting it into this program, you have a negative set of cleavages. So it actually puts all of your substrates into it. So it won't look for other substrates that are in there. And that's kind of how I envision it. I don't know if there's a...
Let me make sure I got it. But are you wondering are these a good mimic of what the substrates are in the pancreas?
Yes.
Whatever it's doing for whatever pathology, is this really sampling
Yeah.
that? I'd say yes, because there's no structure to these peptides. Whereas, right?
Right.
So they're like little bits and pieces of those proteins, but it's diverse enough to be like that book, that if you get a cleavage, if it were wrapped up in some secondary or tertiary structure, then I'd say that's a problem, because some of the endogenous proteins might have to be denatured before they get cut. But yeah, both your guys' questions are really good, because it's how do you control for that? And that we get anything is the way we're kind of going. But can that actually tell us something about some of those substrates? I'd love to be able to go that way. Could we use these to try to hunt, using them as a zip code to search the proteome for maybe it's, I don't know, some kinase or something, something else that's important.
And the substrates were designed at the lengths they are, just so that it basically rules out any like secondary or tertiary structures. So this is our preliminary data. So this is basically showing 2 patients. Patient 22 is undifferentiated invasive pancreatic ductal adenocarcinoma, so the pancreatic cancer is already invasive. Number 26 in white had a low CEA. This is from endoscopic ultrasound. It had a low CEA. It was just blood and epithelium, and had no mucin, which is basically most likely a serous cystadenoma and has no malignant potential. So this shows the FRET substrates here. Each of these shows the sequence of each fluorescent substrate, and you can see that for each one, this represents the activity of the cleavage events. So you can see that they're different. They're pretty different between the two, and that's mainly what we were looking for with this. We just wanted to see differences. And so from here, just to kind of dig in a little deeper and see what kind of classes of proteases were doing this, I took one of the substrates. So this would only be focusing just on this specific sequence. I wanted one they both were cutting it, but still had differences. And then I added protease inhibitors and did the same assay basically. So this is just this standard with no inhibitors. This is a cysteine protease inhibitor, this is a serine protease inhibitor, and this is a metalloproteinase inhibitor. So this basically shows, and this is relative activity, so it's standardized to the control. So you can see in the benign sample, there's a little bit of inhibition in the cysteine, but a lot in the benign sample in the serine protease inhibitor, and a little bit on both with the MMP inhibitor. So this kind of shows us, I was glad to see a difference here in that maybe most of this cutting seems to be coming from a serine protease. And right now this doesn't really tell us much, but it does tell us that there are differences, and that's really the biggest thing that we care about. So this is...
But Dana, just maybe you're going to make that point, but that SPINK inhibitor, that paper that had shown that the SPINK inhibitor is gone, that's an inhibitor of serine proteases. So your data is correlating with that other study that shows genetically, right?
Yeah.
I mean, that's a nice link.
That's true. That is a very good point. Yeah, so in the invasive specimen, you have less.
Yeah, you might think, yeah, we'll see which way it ends up going.
So this is some of the data from the same two patients on the MSPMS. So this is a Venn diagram showing the total amount of cleavages that came from those substrates. And this is from the 4-hour time point. So at the 4-hour time point in the invasive specimen, there were 514 cleavage events, and in the benign there was 144. And then only 77 of those cleavage events were the same. So again, showing pretty significant differences in the amount of cleavages and the amount that they shared that were the same. And so this is one of the biggest things that this technology can generate, and it's basically like a specificity map. So you can think of it as it takes all of the cleavages, and then we'll kind of do some statistics on it. And what it will show you is that you can think of the cleavage event as happening right in the middle here of each one, and then each amino acid position away from the cleavage event. Anything above the line, so the percent difference in the positive direction, will show an increased specificity of the overall protease specificity at that position. So, for example, this is the benign sample at the P1 position, so right next to the cleavage. It has kind of a trypsin-like signature, which has an arginine and a lysine. So it really wants to cut things that have an arginine or a lysine in that position, and that's basically what this is telling you. If it's below the line, it basically says it doesn't like to cut things that have these specific amino acids at that position. And you can see the invasive specimen is just totally different than the benign. So again, good things. And then also, this kind of goes along with that other study in that if there's more trypsin inhibitor, then you're not going to have as much of this activity, which is, we don't see any of that here. And then going back to the carboxypeptidase activity, in our initial MSPMS data, we showed also more carboxypeptidase activity in the invasive specimen. So I wanted to see if we could, again, do it with inhibitors, but this time with the MSPMS, and see if we could get rid of that carboxypeptidase activity. So here, I'm showing you one of the substrates, and that was cleaved basically from the side. And you can see it at these time points. This is, so at 60 minutes we observed this piece, which means that this got cut here. And then at the last time point, we saw both of these. So this piece and this piece, meaning that it's just getting cut sequentially. And when I added this 110 phenanthroline, which is the metalloproteinase inhibitor, we observed no cleavage of that substrate, which basically was exactly the same as the no enzyme control. So just buffer and the substrates with no enzymes at all. And so from this, it seems hopeful that it's an MMP that's doing the carboxypeptidase activity, at least in this specimen. So some future directions that we'd like to do with this is obviously to continue running more patients and get a bigger sample size. I think right now we have about 30 or so specimens to run of varying histologies and pathologies. And then we also have an IPMN mouse model, which is kind of a new thing that actually we can aspirate the cysts and get cyst fluid. So we'd like to compare that and compare it with the human fluid to see if we can actually have a basically comparable, and we can do it at different stages in the evolution of it, which is really great. So we can do it while it's still pre-malignant lesions, when it's not malignant, and then when it becomes invasive pancreatic ductal adenocarcinoma. And then ultimately, the goal would be to take all the specificity data for all of the patients that we profile and have separated them into benign, malignant signatures. And then we can design from these maps, if we get a signature from benign and invasive, we can then design specific FRET peptides that can then be used as kind of like a clinical test that can, within an hour, differentiate a benign from a malignant cyst and know whether or not you need to actually resect it. So I'd like to thank all my mentors and Pathways program. Take any questions?
I have a question. Much like you have with microarray data, there's always the issue of how you're going to analyze the data from a statistical point of view. So have you developed an analysis plan, for instance, in which you've decided a priori how many patients you need to test in different categories, taking into account the multiplicity of the tests to determine how many you're going to need to do to have statistically significant result?
Right now, it's pretty preliminary. Right now, we're aiming for an N of 8 in each category. But there's a lot of different ways that we can do the statistics on it. Most of them I don't even know yet. But some ways are you can actually like compare percent differences here at basically any position. So you can compare percent changes, also numbers of cleavages, and beyond that, I haven't even gotten into the statistics of it yet.
It would be helpful to sit down with a statistician as you're getting into the analysis to design sort of a priori. Because one of the issues comes up when you're doing so many different tests, the statistical significance of one is diluted. So it's helpful to say, is this a preliminary study which is hypothesis generating? Is it something designed to find statistically significant differences? As long as it makes a paper stronger.
Yeah.
Do you think that, just from the literature, I guess, is there a sense that these neoplasm, benign or malignant, or is their secretion profile changing in terms of endogenous signals that stimulate pancreatic secretion? Because in these samples that you're taking from patients or people who've been NPO are likely at kind of a rather unstimulated state. Is there any evidence that maybe the profile could look entirely different